course, the most anticipated application of stem cells is in cell replacement and regenerative therapies. The final goal would be to generate organs that can then be implanted in the patient and to, of course, like replace damaged organs or organs that are not functional anymore. These at the moment, let's say that we are really very, very far from this application. Organs might be generated by like animal-human chimeras, meaning implanting stem cells, human stem cells in animals and then try to develop an organ and then transplant it back. Or a very, very exciting new application is the generation of organs by 3D printing. So the so-called bioprinting.
This is, I was not aware of this technology.
It's very, very, it sounds really cool, but it's very, very far from being like able to be applied. But there is a technology called bioprinting that is one of the most recent medical development in creation of artificial organs. And it would offer, of course, like greater speed and computer-guided precision in the printing organs. The problem, of course, is the more complex the organ is, the more difficult it is to recreate it by 3D printing. There are different levels. I mean, if you have, like, for example, very, very simple structures made by, for example, only one layer of cells, like skin, then I think that the bioprinting is already occurring and maybe it is already quite established, or let's say is going to be soon. But if you have more complex organs like liver, for example, or lungs, then not only you have to that have multiple layers of cells and they're also very, very full of blood vessels. Of course, that creating an artificial organ in 3D printing is really complicated, and we're really, really far from this technology being applied in humans. But definitely there's a lot of research going on, also with collaborations between academia and corporate that is headed in that direction. But of course, that is probably what I would expect we might get in 2050.
And according to you, which are the technological and theoretical or other kind of challenges in your activity?
Mm-hmm. So it is true at the moment that the stem cell research offers really a huge hope for treatment of a wide variety of diseases. But still, there are really a lot of challenges, both from the technical point of view and theoretical point of view. So, for example, although it is true that stem cells can be differentiated in, pluripotent stem cells can be differentiated in multiple different cell types, still there is not many protocols for differentiation in some cell types are not available, meaning that at the moment we are able to differentiate pluripotent stem cells only in a certain number of cell types. And this is also very difficult to scale up, meaning that we are far from being able to produce large-scale amounts of differentiated cells for clinical use. Another really major issue is quality control, meaning that a lot of effort is focused on, once you have differentiated cells from pluripotent stem cells, the population of these newly differentiated cells needs to be thoroughly characterized before you can transplant these cells into patients, because you want to be sure that in terms of gene expression, epigenetic profiles, functional attributes. The cultured cells intended for transplant have not acquired undesired properties. This is actually a known issue for human embryonic stem cells and also induced pluripotent stem cells, because they are known to accumulate, for example, chromosomal aberrations after prolonged times in culture. So chromosomal rearrangement that could give them an oncogenic potential, for example. These have to be avoided. And so the population has to be thoroughly characterized to be sure that the differentiation protocol doesn't induce like mutations or gives the cells undesired qualities. It is also very important to ensure the purity of the population that you are transplanting, because if in the population there is still a very small percentage of undifferentiated pluripotent stem cells that gets transplanted into the patient, these again could have an oncogenic potential upon transplantation.